Got it?
Yep, thank you. Right, okay. Well, thank you very much, Hud, for helping me there, and also for inviting me. It's a great honor to be here, because I'm not a CDG person at all. I really deal with neuromuscular junctions, and I'm not a glycobiologist, so I'm actually really interested to be here, and I'm learning a lot, and I'm hoping to learn lots more as well. So it's a great honor to be here, and thank you for allowing me to come here. So, myasthenic syndromes. So these are genetic disorders, a rare disease, as we're talking about. We've got Mendelian inheritance. They're heterogeneous, lots of variants. The key point here is that actually, if we've diagnosed them and if we can work out what's going wrong at the neuromuscular junction, we can provide symptomatic treatment. And so that's sort of what our ultimate goal is. And additional cases do exist that are as yet undiagnosed. So just to look, and I'll try and bring this up. Okay, so this is a typical myasthenic patient. We're looking at undergoing a myasthenic crisis. And we can see the weakness of the eyes, the ptosis in the eyes, the weakness of the mouth. So we've got generalized muscle weakness here. You can see the arm necessary to try and keep the neck up, and these are life-threatening, and often these sort of myasthenic crises would lead to fatalities if we don't treat them in time. So that shows really just a typical patient. And what's going wrong really? Well, in neuromuscular transmission, the key point is that we've got the nerve and the muscle at the neuromuscular junction. We get the impulse coming down the nerve, releasing acetylcholine. We have to have sufficient release to activate the receptors, and we've got to get a strong enough signal to the receptors to get above a threshold depolarization of the membrane. And so the key thing here is we're looking at a threshold effect. And so that anything that causes a failure to get above this threshold is going to lead to a neuromuscular junction problem and essentially a myasthenic syndrome. And as you might know, most myasthenia is due to autoimmune problem and antibodies attacking the neuromuscular junction, but there's a significant number who have genetic problems. So we're up to now about 22 different genes that can cause these problems, and I don't want to go through them all, just to say that there are lots of different genes affecting the proteins of many different functions at the neuromuscular junction. But how we try to look at them in the clinic in particular is we look at those ones where we've got a defect that's affecting the actual function of the transfer of information from nerve to muscle, such as if you've got too few receptors, or if the receptors are open too long or too short a period of time. And then we've got a whole series of other genes that actually affect the synaptic stability. And in fact, we've probably got about even numbers of patients who have genes that affect synaptic stability or genes affecting the actual function of the signal transfer. So we've looked at a number of these, and in particular, we found a group of patients who we called the limb girdle myasthenia patients, i.e. rather than that effect that I showed you on the video of the ptosis and the face and the eyes being particularly affected, in these patients, what we found was really it's the shoulders and the limb girdle muscles that are most affected. And if I show an example of one of these patients here, this is one of these limb girdle patients. And we found a series of these patients who had mutations in DOX7, but we also found a cohort of patients who didn't have these mutations in DOX7, but looked relatively pretty similar. And sort of features of these patients is the sort of sparing of the face and ocular muscles that I mentioned. We've got usually onset in childhood rather than at birth. We diagnose myasthenia through EMG. And as opposed to the DOX7 patients, these patients tended to respond to anticholinesterase inhibitors. And many of them also, a characteristic you look for, they had tubular aggregates on muscle biopsy, though not all had that. And I just try. Here's another patient. Similarly, here I think you can see more clearly that the facial muscles really aren't affected at all, but she's trying to lift her arms and she's having trouble with that. So that's the limb girdle muscles being particularly affected. So we took this cohort of patients and did, as now is sort of the classic mechanism of exome sequencing. Actually, originally we started with linkage analysis to get our first genes out, but then went on to the exome sequencing, going through identifying variants, looking for similar members of the cohort with similar gene functions. And we came out with a series of genes, GFPT1, DPAGT1, ALG2, ALG14, and GMPPB. And as you might expect in genetics, we have to go and see that we get segregation through families. And obviously, if we've got families, we do segregation analysis, and mutations segregate nicely in a recessive manner through various families that we looked at. And what's going on? Well, typically, if we did analysis of this is actually from a patient muscle biopsy, so we extracted the protein and looked compared to controls, and this was the mutation in ALG2. We see less protein being produced. If we similarly express it in HEK cells, we can again see a similar reduction level of protein. So the mutation is reducing the amount of this protein being produced. And as you might expect, using a figure that's been shown several times today, what we've noticed is that we've got these proteins that are either in the early stages of the end-linked glycosylation pathway or feeding into it. So GFPT1 is obviously feeding substrates into this pathway. So that took us by surprise, totally unexpected, because we didn't think at the time, why would myasthenia actually be due to end glycosylation problems? And the last one, most recent one we've looked at is GMPPB, which actually can give rise to a limb girdle muscular dystrophy and CDG, and also can give rise to a myasthenic syndrome. And this is obviously feeding into the end-linked glycosylation pathway, but also has a very important role in the O-link pathway. So why are these causing myasthenic syndromes? Well, we've actually known for a very long time that the neuromuscular junction is very heavily glycosylated. So here, if we're just doing a binding with lectins, and we've got alpha-bungratoxin, which binds the acetylcholine receptor specifically, so it can be used to identify the neuromuscular junctions. We're binding with lectins. We see the overlap of the binding, i.e. neuromuscular junctions have been known for a long time to be very heavily glycosylated. So just looking at a couple of these, in fact, we see that ALG14 is highly concentrated at neuromuscular junctions. Similarly, we can see ALG2, again, it's quite highly concentrated at the neuromuscular junctions, though that's not true for all of these. DPAGT1 doesn't show such similar concentration at the neuromuscular junctions. So what's going on in our patients? Well, first of all, if we take a muscle biopsy that we've got a neuromuscular junction in and see what's going on, in here we've got a control, we're staining for acetylcholinesterase here, and we can see we get a roughly similar strength staining for acetylcholinesterase. Whereas if we're staining here for the acetylcholine receptor, we see in the control we've got this strong staining for the acetylcholine receptor, but in the patients, it's essentially very weak, and we're just seeing here the acetylcholinesterase staining rather than the acetylcholine receptor staining, i.e. we've got a reduction of the level of the acetylcholine receptors. And if we do an EMG of the neuromuscular junctions, characteristically in the control, you get lots of these invaginations, or what we call postsynaptic folds. And a characteristic of patients who have reduced receptor numbers is they lose these postsynaptic folds. So this is a patient with DPAGT1, and we can see this loss of postsynaptic folds, also indicating to us that we've lost receptors from the membrane. So what is happening? Well, we think probably actually the acetylcholine receptors are being affected, because it's been known for a long time that we've got N-glycosylation sites on all of the subunits of an acetylcholine receptor. So there are 5 subunits, 2 alphas and the other subunits in the mature acetylcholine receptors. The alpha subunit has 1 N-glycosylation site, delta subunit has 3, and it's known that these need to be glycosylated for the receptor to get into the surface plasma membrane to fold up correctly and to get up there. So an experiment done by Kate Balea in our lab with this DPAG-T1 patient, we looked at, if we look at the level of staining we can get or binding we can get of alpha-bungratoxin that's labeled to the receptors on the surface, we see wild type, we get lots of receptors at the surface, or 100%. If we put in tunicamycin, which is an inhibitor of DPAG-T1, we reduce receptor expression. If we put in excess DPAG-T1, i.e. cDNA, that we then transfect into the cells, we can restore the level of surface binding that we get. And if we're looking at the actual glycosylation of the receptor, we see the delta subunit quite heavily glycosylated, or it has 3 glycosylation sites. If we put in the tunicamycin, we've got the loss of this glycosylation. If we put back in DPAG-T1, then we can regain our glycosylation and we regain the surface binding. So we think glycosylation of the receptors probably is playing a role. And if we do siRNA experiments where we knock down other of these proteins that we've been looking at, GFPT1 here, we knock down the GFPT1, and we reduce the level of surface expression of the AChR. And we can do a similar experiment with our ALG4 mutation, where we see a reduced level of ALG14, and so that's expressing it in HEK cells. And if we then do an siRNA experiment, again, we see not actually a dramatic reduction, but a reduction in the level of surface alpha-bungratoxin binding that we give. So we think we've got a molecule here that actually is particularly sensitive to effects on the N-linked glycosylation pathway, and that's what's giving rise to our problems. And so overall, what we identify is that we've got this N-linked glycosylation pathway, and it's affecting the neuromuscular junction. We think the acetylcholine receptors are particularly affected, but it's probably, we know that the junction's very heavily glycosylated and other proteins are glycosylated. So we're likely to be affecting other proteins, but clearly the acetylcholine receptor itself is one key player. And I'd like to say, can we do anything about it? And I'm cheating a bit here because this actually isn't one of the glycosylation proteins. It's a deficiency patient protein. But we can do the treatment exactly the same as I'll show you here and have essentially the same effect with these glycosylation protein patients. So this is a little boy. He essentially can't weight bear and was sat in his wheelchair for about, well, a good 12 years after this video was taken. If we then apply the appropriate therapy for him, shown here, this is him being able to get up out of his wheelchair and walk independently. And it's quite a nice story here because he was essentially, very disabled. His mum had to get him in and out of bed and transfer him in the morning and back. And he was hoping to go to university, and he didn't know whether he could go or not. And his mum was saying, well, maybe he lived in Wales, you could go to the local Welsh university. But subsequently, following that treatment, he's been able to come to Oxford. So he's a bright lad. He's, he has lots of friends at Oxford and is doing really well now and living totally independent of his parents. So we can make sort of life, transforming effects if we find the mutations and get the treatment right. So, I mean, this just shows sort of QMG scores, which is a score of severity for myasthenia that we can use for sort of assessing our patients. And typically here, once following treatment, we see the severity comes down. And these are sort of just arm raises and leg raises and sort of measures that we can use for sort of everyday muscle function that we can see where they're increasing. And this just, I mean, these are 6 patients that we were studying, and we can see we can sort of make quite a considerable difference in their strength and their ability, and it is usually life transforming for the patients. So lastly, I just come back to GMPPB because that's the most recent thing we've identified mutations in that can cause myasthenic syndrome. As I'm sure most of you know, mutations in this gene can also cause a limb-girdle muscular dystrophy and more serious problems. Just at the top here, we've got the mutations in some of our CMS patients, mutations in cases that are causing more serious problems. And we can't really see why one should give rise to a myasthenia and one should give rise to a muscular dystrophy. Moreover, if we look at actually in some of our patients themselves, this is within one patient. And if you do neurophysiology, EMG, which we classically use to define a myasthenia, this is in a single patient, one muscle we're looking at, and we see the decrement, which is the characteristic hallmark of a myasthenic muscle. Same patient, different muscle, no decrement at all, i.e. very muscle-specific effects occurring. And in C at the bottom here, we're looking, this is EMG, looking at myopathy, and you can see the characteristic signal for a myopathy. So in this patient, we've got some muscles that don't seem to be affected at all, other muscles where we've got the neuromuscular junction affected, and other muscles where we've got a myopathy occurring. So it's obviously quite complex, and we don't know what mutation, why mutations, or why patients should be affected in different ways. So just to summarize, we've here broadening the phenotype that we see with myasthenic syndrome. So we've got lots of patients where we're sparing the face and ocular muscles. And normally, if you think of myasthenia, you think of ptosis, you think of eye and facial muscles being affected. So we've got a whole series of disorders where they're not affected. And obviously, that's probably leading to underdiagnosis. We can have onset in adulthood or childhood or late childhood, so variable age onset. In the case of the GMPPB patients, we've got a raised CK, and that's very unusual for myasthenia. And so therefore, that sort of is marking this out for us, so that if we see a patient with raised CK, we'd almost immediately look at GMPPB, and probably that's indicative of the myopathy rather than the myasthenia. As I've just mentioned, we've got variable severity with overlapping myopathies, variable neurophysiology, but the patients that have the clear myasthenia are still responsive to pyridostigmine and salbutamol and ephedrine, which we use a combination of those to actually get the best response because we tailor our treatment for our patients. So just putting here, I mean, so these are rare diseases. We've got about 500 cases that we study in Oxford. The main cases are there, but we've got this expanding group here that is expanding all the time with what we're now finding is mutations in the glycosylation pathway. And just to finish, so essentially what we've got aberrant glycosylation. We've got this group right down the far end, which we would think of as the very mild end of the spectrum. So they're not, I mean, they're affected, but not nearly so seriously affected as if you've got these multi-system disorders, or in fact, most of the limb-girdle muscular dystrophies are more seriously affected than our patients. Obviously, there's overlap here. There's a spectrum. We don't know why you can have the same mutation and seem to get very different phenotypes coming through. And we think we're having the acetylcholine receptor affected, but actually we're probably also getting other proteins at the neuromuscular junction being affected to give rise to these congenital myasthenic syndromes. And so I'll stop there, but just like to thank the members of my lab here who do most of the work. And I'll leave it there. Thank you.
Questions? Yes, please.
I can speak loud. So my question was the salbutamol and ephedrine treatment of these individuals with milder and focal issues of glycosylation of the acetylcholine receptor, could that be extrapolated to children with more severe CDGs in terms of their myopathy?
Okay, sorry, so I haven't quite filled in properly. So what we do is we're treating the neuromuscular junction disorder, and we think we're having both effect on the receptor level and on the synaptic structure stability. And we essentially, for the receptor level, we treat with pyridostigmine, and also for the stability, we treat with the beta-2 agonists, ephedrine or salbutamol. So in fact, for these patients, we treat with a combination of both those together that has the best effect. And we also have a third treatment with 3,4-DAP that we sometimes use as well. We've only seen mutations at the very early stages of the CDG pathway. So I don't know whether that's because the myasthenic component is lost in the sense of investigations further down the pathway, or that actually the effect on the neuromuscular junction is only seen through the effects on the very early stages of the pathway. So I don't know, is the ultimate answer to that, whether these therapies that are symptomatically treatments for the neuromuscular junction, so they're not treating the myopathy or the other components that are going on. So they're just effectively increasing the signal transmission at the neuromuscular junction. So I don't know whether those would affect other patients with, other genes involved.
Actually, maybe just answered my question. So I was curious if you'd seen any myasthenic presentations in GPI anchor disorders, since acetylcholine is a GPI anchored protein, and we do see ptosis and some other things in other types of CDGs.
I haven't seen that in our patients. I mean, ptosis can occur in, quite a few disorders, so you can get it in mitochondrial disorders, et cetera. So, and obviously muscle weakness, hypertonia is common in lots of things. So, we need for us to have an EMG saying decrement, really, for us to say, this is a myasthenic disorder. And then that's when we say our treatments are going to work.
I think this is probably one of the more amazing pieces of glycobiology that we know about, to see such a divergence in the symptoms. And as you point out, it's in the early part of the pathway, and there's a few other genes in there that might actually be candidates, and maybe we'll run into them. But since we've already heard about ManNAC therapy, we talked about the potential of Manos therapy, I'm just wondering, because 3 of the defects actually involve the incorporation of glucanac. I'm not saying you go try glucanac, but it makes me wonder as to whether there might be any possibility that a little extra push of glucanac.
We did try that in the GFPT1 patients, and we didn't. I mean, we've got a patient who's still taking it, so it might be having a modest effect. We haven't seen a dramatic effect, but so it hasn't been dramatic, but there might be a mild effect.
Okay.
So we have some.
How much are you giving?
I can't remember. You'd have to go back and I'd have to refer to the notes, because I'm not actually a clinician. I just advise on what the clinicians do.
So I just want to, make sure that I understand correct. So those patients does not have any like a transferring underglycosylation. So it's only the
no, sorry, so I haven't been clear. Some of them, you can, if you look hard enough, you can see a modest change in transferrin, but most of them don't. So I think we've got a couple of DPAG T1 patients who can be picked up by transferrin assays.
I think the most of the protein, the mutant, is actually really functional, but maybe a little bit less activity, which causes a very specific effect on the CNS.
So we haven't got any patients who've got a double knockout. We've got some patients with a null allele, and then missense on the other allele, which either modestly reduces or reduces expression levels, or maybe having an effect on the catalytic activity of the molecule. So, yeah.
Right. So thank you, all three speakers, and I think we go to coffee break.